Hello and welcome to Nikolai's genetics lessons, and today I have prepared to raise simple questions for you. You probably know answers, but still I recommend you to watch this video to the end because I hope I can still give you some interesting information that you probably didn't know before, and that might help you on your exam. The first question is, the sugar found in DNA nucleotide is, and you have to choose the correct answer, while the sugar found in RNA nucleotide is, you have to choose the correct answer. And you would be able to choose the correct answer if you would understand the difference between deoxyribose and ribose. And the only difference is that ribose has a hydroxy group on the second carbon, so we have carbon 1 here, carbon 2, carbon 3, carbon 4, and here we have carbon number 5. And in deoxyribose, as you see, deoxy means that on the carbon number 2 it misses oxygen. So this is the only difference, and also you may guess that D here stands for D and DNA, and R in ribose stands for R in RNA. So the correct answer would be the sugar found in DNA nucleotide is deoxyribose, while the sugar found in an RNA nucleotide is ribose, and this is answer B. And by the way, do you know the difference between glucose, sucrose, and pentose sugar that we can find in deoxyribose and ribose? The difference is that glucose and sucrose are 6 carbon molecules and pentose 5 carbon molecule. So the structure of the glucose would be like this, with 6 carbons as you see here, and pentose would look like this with 5 carbons here, but here we wouldn't have carbon. As you see, this is oxygen here. The 5th carbon would be here. This structure in geometry we call hexagon, and this structure we call pentagon, and penta in Greek means 5. So glucose and sucrose our organism would use as energy source, and pentose would be produced as a result of digesting of the glucose, and this pentose would be essential part of the ATP. This pentose is not essential to be present in a food because our bodies can produce this sugar. And according to this picture, as you see, deoxyribose would be derivative of the ribose. And now we can move to the next question. The structure of DNA is, you have to choose the correct answer, while RNA is generally, and here is answers to choose from. I hope everyone knows the answer to this question, that DNA is double-stranded, while RNA is single-stranded. Prokaryotes, because they don't have nucleus, so when messenger RNA would be transcribed, simultaneously it also would be translated. But in eukaryotes, messenger RNA or any other RNA, structural RNA, like what we can find in ribosomes or such adapter molecules as transfer RNA, usually represent complex structures with some fragments that would be that would make a double-stranded RNA in such places where RNA would have a fragment that is going to be self complementary. So in eukaryotes, RNA usually found in such form and not in linear form like in bacteria. Because in bacteria, messenger RNA, for example, would be transcribed and translated at the same time because it doesn't have nucleus, but in organisms that has nucleus, in such a cells, transcription and translation would be separated in space and in time. So as you see, the correct answer would be answer A. The structure of DNA is double-stranded, while RNA is generally single-stranded. So as you see, this video covers two main differences between DNA and RNA, but actually there are three main differences. Do you remember which one is the third difference? And the third main difference between DNA and RNA would be that DNA has bases adenine, guanine, cytosine, and thymine, and RNA has bases adenine, guanine, cytosine, and uracil. Uracil is just structurally almost the same as thymine, but methyl group. So in RNA, thymine would be substituted with uracil. This is the third difference between DNA and RNA. And one more extra interesting piece of information for those people who watch this video to the end. There actually exist double-stranded RNA. Some viruses has the genome as double-stranded RNA. We call this double-stranded RNA viruses. Some also can be single-stranded RNA viruses. That strand can be positive, negative, and also some viruses also can be single-stranded DNA viruses and double-stranded DNA viruses. So as you see, there are a whole range of different possibilities. And this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day. Thumbs up if you like this video. Please share this video with your classmates and see you in the next video. Goodbye.